So we all know digestion of carbohydrates starts from the mouth. So we have the salivary gland producing salivary alpha amylase. Now this enzyme, what it does is it targets the 1,4-glycosidic bond in amylopectin amylose, forming maltose, maltotriose, and alpha-limidextrin. Now, since food doesn't stay that long in the mouth, it doesn't do much. The main work is done by the pancreatic amylase in the duodenum. Now, it does the same work. Now, after that, we go to this place. So we have the enterocyte brush border enzymes. We have lactase, maltase, isomaltase, and sucrase. These two are related together. Then for isomaltase, it targets the alpha-limidextrin. Now, it targets the 1,6 bond, the alpha 1,6 bond, which is this bond here, this branching. That's what it targets. And as a result, you absorb your monosaccharides and eventually enter into the hepatic portal vein. Now, we all tend to think that only carbohydrate digestion starts from the mouth, but fat digestion starts from the mouth too. But it doesn't spend long in the mouth, so the main one is the stomach starting it. So, lingual lipase and gastric lipase breaks down triglyceride into monosaccharide and fatty acids. Now, when you get to the duodenum, the pancreas secretes bile and pancreatic lipase. The bile emulsifies the fats, and pancreatic lipase breaks down the triglyceride into monosaccharoid and fatty acid. Now, the pancreas also produces other enzymes like phospholipase A2 and cholesterol ester hydrolase. Now, eventually, you get micelles. And in this micelles, you can usually see the vitamins entering into the fats, which is vitamin A, D, and K. Then, bile salt leaves, and these micelles enter into the intestine enterocyte cells, where they're taken to the smoothie R and converted back into triglycerides, cholesterol, and cholesterol esters, and eventually taken to the rough glycosylated column for more modifications before leaving through the highly specialized lymphatic capillaries known as lacteals and go to the liver. And some kinds will go into the skeletal muscles and adipose tissue. Now, for protein, it's much more straightforward. So, in the stomach, pepsinogen is released. Now, around 1.8 to 3.5 pH, pepsinogen turns into its active form, pepsin. Now, pepsin breaks down large polypeptides into small polypeptides. So, entering into the duodenum, the pancreas releases trypsinogen along with other coenzymes. Now, trypsinogen is converted to trypsin by enterokinase. Now, trypsin converts these coenzymes into their active form, chymotrypsin, carboxypeptidase, and elastase, and they all turn the small polypeptides into tripeptides or dipeptides or amino acid. Now, for the amino acid part, it is mainly the carboxypeptidase and elastase that turn the small polypeptides into amino acids. Over here, we have the heteron brush border enzymes. We have the dipeptidase and the tripeptidase. So, both of them turn dipeptides and tripeptides into amino acid. Now, not all of them is converted, so we have some dipeptides and tripeptides moving into the enterocytes along with the amino acid. Now, all of them move into this transporter and go into the hepatic portal vein.